Here we go. Today we're going to talk about this paper, Enhancement of Zika Virus Pathogenesis by Pre-existing Anti-Flavivirus Immunity. This was published in Science 2017. I have the ugly version of it, the one from, I think, PubMed. The prettier versions in Science. But this is a really good paper, I feel like, and it's an easy paper to read. It's an easy paper to understand, so I feel like this is a much better paper to lead the discussion on antibody-dependent enhancement as opposed to the other paper, which was originally assigned. Okay, so the premise of this paper is, which virus is this paper concerned about? Zika. Or in the paper, they'll say Zika. And this is not 5, this is Zika virus, Zika virus. That's the one they're concerned about. So this was in 2017, just after the explosions in Brazil of Zika virus, and it appears, it's probably going to be a bad Brazil. Something like that. There's Florida, Texas. There's an explosion of Zika virus, and it spread up, and we are in terms of like the United States, southern Florida, southern Texas, and parts of Alabama, Louisiana, Mississippi are in the sort of like the region of which Zika can spread. So we hear a lot about Zika. It's a virus that we worry about. And so the context of this is that as the Zika virus was spreading and kind of like moving northward out of Brazil, it was spreading into regions that were already endemic with other flaviviruses. So the other flaviviruses, which we now should be kind of familiar with, one of which is West Nile virus. We have West Nile virus all over the United States. It's a flavivirus related to, in a similar sense, to Zika. And also dengue virus. Dengue virus, I think in the paper you see it. You see den virus. Dengue virus. So the Zika is expanding its range into regions that are already endemic with these other flavies. Okay. And the big concern, what's the big concern with that?
That if you've already had, especially dengue or West Nile virus, it makes it easier for it makes the Zika more like intense almost if you get it.
Yeah, so I'm going to repeat what Lauren said because nobody could hear. What Lauren said was that it's possible that if you have had dengue or West Nile virus, that it could make the Zika infection that you get later even worse. So this is the phenomenon that we were starting to talk about last session, which we call ADE, or antibody dependent enhancement. And just again, a brief review. What happens is, as your body is infected with viruses, your body produces these antibodies, and these antibodies are what are used by white blood cells to recognize pathogens and destroy them. And if your body has antibodies that dengue, we'll do dengue, that's the best example. Let me erase this. Okay. The most famous example of this is dengue, where if you have been infected with a dengue virus, let's say dengue 1 in the past, maybe when you were a baby or maybe when you were younger in life, your body is going to have antibodies for dengue 1, and later if you get a different strain of dengue, let's say dengue 2, that dengue 2 virus can see these antibodies and use them as like a launch pad to infect these white blood cells, and then it can reproduce. So in the first infection, the antibodies are assisting your health and helping it and kill off the infection, but then a second subsequent infection with a different strain, they can be a launching pad for that virus to invade your white blood cells. And it makes the pathology very, very bad. And that's how you get the dengue hemorrhagic fever and dengue shock syndrome, which kills people. So the context of this paper is the big question they're wondering about is since we know this phenomenon of antibody-dependent enhancement happens with dengue virus, well, there's other viruses that are very closely related to dengue virus. There's other flavies like Zika and West Nile virus. And so there's a question of, well, if you've already been infected with a dengue or you've already been infected with the West Nile virus, is it possible that the antibodies produced by your body against these viruses could produce antibody-dependent enhancement for subsequent Zika infections in the future? So it's essentially like a big question, like, do we have to worry about Zika infection becoming more and more pathogenic because people in the United States or in other countries have already been exposed to other flavies like West Nile virus and dengue? So that's sort of like the big question of this paper. Now, also, this applies to like a Zika vaccine in theory. Like if people are trying, that was a big rush, was people wanted to produce a Zika vaccine. If this was true, what would be the issues about or surrounding the Zika vaccine? If you saw antibody-dependent enhancement with Zika virus, what might be an issue from a future Zika vaccine? In the chat. If you get the Zika vaccine, it could make dengue worse. Yeah, you could have the reciprocal of literally what I just said, the reciprocal argument. Whereas if they produce a good Zika vaccine and everybody goes out and they get the Zika vaccine, then maybe later in life you could have the reciprocal of what just happened, where if you have really good antibody response in your body against Zika virus, and then maybe later you get West Nile virus, or later you get a dengue virus, then maybe now you're subject to dengue hemorrhagic fever and you die, because in the past, perhaps you've been exposed to that flavivirus vaccine, which produced the antibodies against the Zika virus. So this is actually like a legitimate concern that needs to be studied, and that's what this paper is exploring. Okay, so in the guide, some of these sentences are worth, I pulled some of these sentences from the paper. And some of these sentences are worth like going over, like, what do they mean by this? So here's a sentence from the paper. ADE is primarily mediated through the engagement of IgG antibodies with cell surface FC, I think that's gamma RS, cell surface. Oh, okay. What does this sentence mean? This one. What is it saying? ADE is mediated through engagement of IgG with cell surface, this thing. What does that mean? Do you understand that, Ifan?
So the mechanism of the IgG bond with the surface receptor is
what is IgG? It's your antibody protein. Okay, so let's first define what is in this sentence. Okay, there's a key word which is IgG. That's the name of a big antibody protein. That's the name of the antibody protein. Okay, so when I draw like this Y, it could be the IgG protein. Okay, so they're saying antibody-dependent enhancement is primarily mediated through the engagement of IgG antibodies with cell surface receptors, FC, whatever, you don't need another name. What they're saying is there's a receptor in the surface, in the bilayer of these white blood cells, these leukocytes, and there's a protein in that surface, and it's got this strange name, FC gamma Rs, probably FC gamma receptor. So there's an FC gamma receptor, and that receptor binds to IgG. Okay, so there's these cells, and they pin themselves with antibodies, and the antibodies have interaction with this surface receptor. And the antibody-dependent enhancement is the mechanism of that is mediated through this sort of like interaction. Does that make sense? So like the virus is capitalizing on this interaction to invade the cells in antibody-dependent enhancement. That's what that sentence means. So that's an important sentence to understand. And you don't necessarily, it's not important in the sense that you understand, like, remember that name, but it's important that you remember or know that there's like receptors on the surfaces of white blood cells, they bind to antibodies, and this antibody-dependent enhancement effect is mediated through those interactions. And you should remember IgG. Okay, so how are they gonna, what's the approach of this paper? So let's start thinking about some of the approaches that they take to investigate this question of whether there's going to be antibody-dependent enhancement. How would you guys address this question? Or how do they address this question?
They used recovering mice that had these what's now or Zika infection? Not Zika, but
Not recovering mice.
well, because I looked up what convalescent was.
Mm-hmm. So let's start with that. Convalescent plasma. This is a big word nowadays. Have you heard that before?
No.
In the context of COVID? You really?
With COVID means, or something.
So convalescent plasma, somebody take a guess. What is that? Excellent. Good work, Ifan. Ifan says, if a person is infected with a virus, let's say Zika virus, that person, right, has blood flowing through their system, and in that blood system is produced antibodies that recognize the Zika virus. Now imagine a scenario where this person fights off the disease, okay? They fight the disease. What happens if you fight off the viruses? What happens?
You have evidence that you won the antibodies.
And you're immune for the rest of your life for most of these. If you get yellow fever and you get it once and you fight it off and you don't die, you are immune for the rest of your life. So if you fight these off, your body has adaptive immunity and you are immune. Okay, so the idea behind convalescent plasma is a convalescent is a person who has gotten a disease in the past and fought off the disease. They're now immune. So there's something in their blood system, namely these antibodies, which are now protecting them for the rest of their life. So the basic idea of convalescent plasma is, let's say you have Sally and Joe, and Sally got Zika virus long ago. She has these valuable antibodies that can save somebody's life. So a blood bank might take what's called convalescent plasma. That means they extract Sally's blood. They put it in a tube. You can centrifuge it. When you centrifuge it, all the blood cells go to the bottom, and what's left in the supernatant is called the plasma. That plasma has these antibodies, okay? So now what you can do with that convalescent plasma is you can take that plasma, put it in a syringe, and inject it in Joe. If Joe gets Zika virus and he's very, very sick, inject him with Sally's plasma. Now all of a sudden you bolused him with IgG antibodies that recognize the Zika virus that can help his immune system fight off the virus. That's the idea of convalescent plasma. The reason I said it's a Bing word right now is because convalescent plasma is one of the few treatments of COVID that has like statistically significant beneficial effects. So people are collecting COVID convalescent plasma and using it as treatment. But you can do the same thing with any virus in theory. Okay, so the idea behind the experiments in this paper is, let's get people who have been infected with these different flavies, West Nile virus and dengue, and let's extract their convalescent plasma. I'm just going to say CP. Let's extract their convalescent plasma, and then we can do these experiments with it. We can test it to see, we can test for binding We can test to see if those IgG antibodies in that convalescent plasma can bind and recognize certain proteins on the Zika virus. We could test for virus invasion assays. So like, are the viruses invading the cells? Are they replicating in the cells? We can measure that. We can measure death. And they measure death in a mice model, which is probably what caused a little bit of confusion. But the convalescent plasma that they're drawing from is human plasma, if I read the paper correctly. So that's kind of like the gist, is let's test some hypotheses utilizing this convalescent plasma. And let's see if the convalescent plasma, the antibodies in there, let's see if they cause antibody-dependent enhancement. Okay, so I have another sentence that I pulled out that I think is worth discussing to make sure everybody understands what it is. If I don't freeze my computer here. In all flaviviruses, the envelope protein E is a primary target of the neutralizing antibody response. And recent studies show that the Zik5E protein is highly structurally similar to that of dengue virus and West Nile virus. What does this sentence mean? Somebody explain to me this sentence. Very good, very good. So Dee said that when, let's say this is the Zika virus. When Zika virus gets in the body, it has these proteins on its surface called E proteins. They're in the envelope. And the first thing that those antibodies recognize, the first thing is the E protein. Okay, so translate this sentence. First point is that the IgG antibodies recognize the Zik5E protein. That's the protein that the response is mounted against. Obviously, the antibodies are not going to bind some protein that's inside in the middle of the virion, right? Does that make sense? It's obviously going to be the outer surface protein, okay? The Zik5E protein is that protein that mediates that interaction. Okay, that's the first half of the sentence. What's the other half of the sentence? This one. Why is this significant? E proteins, in a good way to say this short and succinctly, is E proteins and flavies are conserved. That means if you do an alignment, like some phylogenetic tree, you look at the amino acids and they're lined up, and you have E protein from Zika virus, and you have E protein from West Nile virus, and you have E protein from dengue virus, they're pretty much the same thing. They're pretty much the same thing. They're structurally similar, is what the sentence says. Okay, there's going to be some differences, but they're structurally similar. What does that mean? Now translate why that's important to what we've been discussing the whole time. If the proteins are structurally the same, what does that mean? It means it's something that recognizes this, probably also going to cross-react. There's a term for this called cross-react with the E protein from West Nile virus. It's probably also going to cross-react with the dengue virus E protein. Okay, so there's probably going to be cross-reactivity of antibodies. And in the context of antibodies and vaccines, this could be good or it could be horrible. It could be good in a sense that maybe you get a vaccine for Zika virus and you're also protected against West Nile virus and you're also protected against dengue virus a little bit. Maybe that could be the scenario, or it could be opposite of that. It could be a scenario you get a Zika virus vaccine and now you're subject to West Nile virus, dengue virus, antibody-dependent enhancement invasion, and you could die. So again, that's sort of just an important sentence. You have to understand that sentence to understand the context of this paper. Okay, another sentence. Several studies have evaluated dengue virus specific monoclonal antibodies or a limited number of immune plasma samples for activity against Zika virus. collectively, these studies show that in vitro, some dengue virus antibodies are cross-reactive. So we just described what that would be. They're saying people have looked into this, and that is the case. That is the case. People have looked into this, and antibodies that are produced against dengue virus are cross-reactive to Zika virus. Okay. And then they say, however, that sort of that cross-reactivity didn't translate to enhancement of Zika virus. They're saying, however, enhancement of Zika virus infection by dengue virus antibodies was not observed. So what they're saying is, in the past, people studied this, and remember how we said there could be two scenarios here, one where it makes it better, one where it makes it worse, and they're saying the second scenario where it makes it worse hasn't been observed in the past. Okay, so that's what they're saying. Important context. Now I want to point out this. This is a hard study to do. Why is this a hard study? Many things that make studies hard to do are sort of like super valuable resources. What's like the valuable resource here that not everybody in the world has access to? Everybody in the world has access to E. coli. Everybody in the world has access to mice. Everybody in the world has access to cell lines, human cell lines. What's the valuable resource here that is sort of makes this study a high impact paper that makes it difficult? Not everybody could do this. The plasma. How do you go about getting plasma from, I don't know how many, tens of thousands, maybe hundreds, I don't know how many people, but probably thousands of patients. And it's not just patient plasma that they need. It's a special type of plasma. It's plasma from patients who have in the past gotten West Nile virus and then fought it off, or gotten dengue virus in the past and fought it off. And especially with dengue, I'm sure they're looking at specific like strains. Like it probably couldn't just be like any old strain of dengue. They're probably like trying to standardize it, I would imagine. So like that is not an easy thing to do, to just be able, oh, I'll just go find some humans and I'll take their blood, and like that is not something that is easy to do. Okay, so this requires sort of a lot of coordination with governments, with hospitals. It's not an easy study to do, which is probably one reason why it's in Science. Okay, it's hard to do. And so they got this plasma. This is where the sentence where they talk about where they got this plasma from. We evaluated convalescent plasma. Okay, so from 141 people, 141 immunoglobulin positive dengue virus infected patient donors, and 146 positive West Nile virus infected blood donors. Okay, so they got not thousands. Okay, so that was incorrect, but 150 people who had been infected with dengue and 150 people about who had been infected with West Nile virus. And they got those from blood donations in Puerto Rico and the United States. Okay, so they have this valuable resource, which is this special plasma. Okay, so now imagine their special resource is this plasma, which it's gonna have antibodies for West Nile virus. It's gonna have antibodies for dengue virus if it's either A or B. What's gonna be the key negative control that they're always gonna be comparing these things to? Key negative control. Anytime you do an experiment comparing either this or this, A or B, to something, what's going to be that something that they have to compare it to? A healthy person who has never been infected with either West Nile virus or dengue. Okay, so it's going to be a negative, so that would be negative West Nile virus, negative dengue virus plasma. Okay, so they also need a key negative control, which is somebody who's never been infected with these before. There's also another control, another negative control. What's the other negative control? Usually every time they do an experiment looking at this and this, they definitely compare it to this, but there's some experience where they compare it to something else. What's that something else?
Phosphate buffered saline.
There are some times where they compare things to PBS, phosphate buffered saline, but that's not the one I'm looking for. What I'm looking for is sometimes they take plasma, which is from patients, it's convalescent plasma, from patients who have either been infected with West Nile virus or dengue, and they do a removal of the minus IgG. GG. How is D going to be different from A or B? Like what phenotypically, what would we expect would be the difference? They're not going to bind to the virus, because if that interaction is mediated by the IgG antibody and the IgG antibody is no longer there, it should be a complete negative control. You see how that's sort of, it's like a more robust negative control. Does that make sense? And so by this control, by comparing to this control, any effect that they get, they're going to be able to prove that it's directly attributable to the IgG interaction if that phenotype goes away when IgG is not there. Does that make sense? Those are two key controls. You have to understand those key negative controls before you start looking at like what they're comparing things to and trying to understand their data. Okay, one more final sentence to discuss. Let me just look at this quick. I don't know why that's there. I don't care about that right now. I don't know why that's there. Okay, so let's start looking at the data. Okay, let's look at the paper. And let's just start at figure 1. This paper is quite easy to understand. If you just take it slow and look at the figures, it's really easy. Okay, so let's zoom in on. So this is figure 1A. Okay, y-axis. Look at the y-axis. Zika E protein ELISA. Okay, so let me first explain ELISA. What is ELISA? Have we ever heard of ELISA before? No?
Yes.
What, can you explain it?
The enzyme linked immunosorbent assay.
So yeah, it's enzyme linked immunosorbent assay. Yes. It's essentially like a thing that tests binding. And usually it's specifically testing binding of antibodies. Okay. I mean, anytime you see ELISA, you start to think of antibodies and pathogenesis and binding. And what they do in general, like there's different ways you can do ELISA, but in general, like, okay, you get like a 96 well plate. So these plates have a bunch of different wells, on and on and on and on. And in each of these wells, okay, if you were to zoom in and look at these wells, each of these wells, the plates are special. They manufacture special plates that are bound to antibodies, okay? And whatever that special antibody is, whatever that thing is designed to recognize is going to be the thing you're searching for. Does that make sense? So like ELISA is a way to detect if something is there. ELISA detects presence of what we call antigens. Antigens are things that stimulate the immune response. They're proteins that cause the production of antibodies to be produced. So in this case, what's the particular antibody that they're interested in searching for? That's the antibody. What's the particular antigen that they're interested in searching for? So you're right that this, well, I'll describe this in a second, but you're right, it would involve IgG, but what would that IgG be searching to bind for? Not the receptor. What? What part of the virus? We talked about this. The E protein. The E protein is going to be their target. The E protein of these flaviviruses is their target. Okay, and what they probably do is probably what's called a sandwich ELISA, which is they probably have in the well. I'm just guessing here, but I'm going based off my sort of what I know about ELISAs and my knowledge of molecular biology. In each of these wells, they probably start with an antibody, and that antibody probably recognizes it. I'm thinking, how would they do this? Because what they're actually doing is I feel like they're pulling out the antibodies from the plasma. So they could have in theory, one thing they could do is they could have an antibody which recognizes IgG, And then what they could do is they run the blood, the plasma, they run the plasma over these wells, and then that antibody is going to bind to the other antibody, the second antibody, which is the IgG, which recognizes the Zika virus, and then they could detect whether or not it has the Zika virus in there. Let me look at the y-axis. Because what they're actually measuring is the E protein. The other way they could do this is maybe they don't do that double antibody thing. The other way they could do this is they could literally just buy a plate with some antibody that's known to pull out the Zika 5 E protein. But what they're showing you on the y-axis is the quantity of E protein in the blood. Okay. Hang on, I'm thinking this because this is measuring binding. Here's the negative control. So these would be from patients that have not, oh my God. Can I draw on this? That's going to do some signing. Oh, I don't want to do this. Oh, my whole lecture's falling apart. My whole life is falling apart. Oh no. Okay. I just want to close this. I'll just look. Okay. So control is going to be they're running over plasma from people. Hang on. Okay, I got it. Well, it's in my head. Let me draw it out. So what they do is they take plasma from the patient. What they want to extract out is the IgG from those patients, okay? So they're going to run the plasma over the 96 well. There's going to be something in the well that grabs the IgG, okay? So then after you run the plasma over those 96 wells, you've got 96 wells filled with the IgG from those patients, okay? And then what they do after that, so this is step one. Step one is collect the IgG from patient plasma into the ELISA setup, okay? Then what they do is step two, run Zika E over the 96 well plate, and they can quantify how much E binds. So if the patient has antibodies that will bind the E protein, that y-axis will shoot up. And so what they're saying is in patients that had been infected with the dengue virus, their IgE proteins are binding and pulling down the Zika 5 E protein. You see it goes up, okay? So the dengue virus antibodies, IgG antibodies that recognize dengue virus are also recognizing the E protein. Ditto with West Nile virus, but not as tight binding. Does that make sense? Now the next experiment, B. So A is testing, is there cross-reactivity? The answer is yes. B is testing, B is a different setup. This is no longer ELISA. B is testing fold enhancement of what? Did you guys catch that? So the y-axis is fold enhancement in figure 1B. What does that mean? What is that related to? Who read the paper? Fold enhancement of what? Yes, Zika infection in what type of cell? Human cells. Some strange number, but yeah, human cells. So they have human cell culture. So imagine this is a culture of human cells, okay? And what they do is they take virions. So step one, take Zika virions. You should know what I mean when I say virions. It's a particle. It's an infectious particle of Zika virus. Take Zika virions, 2, mix them with the convalescent plasma and then throw the mixture on the cells. Throw the mixture on the cells. And then compare that, can count, what they're counting is how badly the cells are infected with virus, how badly the cells are infected. They're probably quantifying replication of virions. So if virions replicate in the cell, you'll be able to see that. You'll be able to quantify that, okay? And the fold enhancement is in comparison to a negative control, okay? So in the controls, there's no enhanced infection of Zika virus when they mix that plasma in. If you mix the plasma with the virions and the plasma was derived from a patient that had been infected with dengue, it enhances Zika virus's ability to replicate in those cells that they throw it on. Same thing with West Nile virus, but not as much. Do you get that? Like, does that make sense, that experimental setup? Okay. And these are just statistical correlations. If I can, why can't I slide over? There it is. These are just statistical correlations. You see how there's like a line here? There's a line, there's a line. Essentially what this means statistically is more IgG equals more infection with Zika virus. There's a correlation, okay? So that's the gist of figure 1. So what's the conclusion? The antibodies for West Nile virus, the antibodies produced by the patients that recognize West Nile virus and dengue virus also can recognize the Zika virus, and they also enhance the infection of Zika virus in human cell cultures. That's important, in human cell cultures. Okay. Next figure. Glad we could work through that. Sometimes these can be challenging. Okay, A. So I'm pretty sure these are now, again, yeah, they're still doing human cell culture. So they're still doing the experiment style where, okay, so we're on figure 2. Figure 2. They're still doing the thing where they're taking virions, mix them with convalescent plasma, and then throw that cocktail on cells, and then quantify number of infections or like number of virus. They're still doing that. They're still doing exactly that experiment in human cell culture, okay? But now what they're showing you is these are diluting the plasmas, okay? So the y-axis is just like before, fold enhancement, but the x-axis is dilutions of the plasma. So you would expect that the effect would go away if you start to dilute the plasma. Does that make sense? So all these will merge or converge towards the control value as you're diluting the plasma, okay? So that's sort of the x-axis. Now what's important in each of these figures is these comparisons. The comparisons of the dotted lines to the solid lines, okay? So control plasma has not been infected with the virus. This is like baseline infection levels of the Zika virus. Does that make sense? Baseline, okay? And this is control IgG. What they're doing here is, so this figure in figure 2A, they split this experiment apart. When I said they're doing this, they split it apart. In the first half, they're doing mix with plasma. In the second half, they're doing mix with just IgG. And you would expect that if IgG is the cause of antibody-dependent enhancement, 2 will look exactly like 1. Okay? That's what we would expect to see. So here's the infection of Zika virus, the red solid line. If the plasma was derived from a patient that had West Nile virus, it's higher than the control. So you're seeing enhanced infectivity. And if you don't use the plasma, but you use the extracted IgG, it looks the same. It's the dotted red versus the solid red. They look the same. And the same thing for blue. Dotted blue looks the same, pretty much the same as dotted blue. So what this is saying is essentially this effect that you're seeing of enhancement is because of the IgG protein. Does that make sense? Yes? Okay. B. Now they're doing that key negative control that I talked about. Now they're comparing the plasma as normal, the plasma. They've done many of these experiments with the plasma, but now they're doing this key negative control where they're comparing it to that same plasma with the IgG antibodies purified out. So they've subtracted out the IgG antibodies. Now in this case is exactly opposite of what I just told you. In this case, would we expect them to be the same or opposite? The results. Opposite, because this in the plasma, their whole argument is that in the plasma, the key reagent is IgG. Okay, they're saying the reason that you get this increased infection in the blue and the red is because IgG in the plasma. So obviously, if they remove IgG from the plasma, it should look just like the negative control. It should be baseline. So that's what they've done. They've done that experiment. Okay, so they've actually proven that it's the element. IgG is the element in the plasma that's causing this effect. So these A and B are kind of like reciprocal experiments. Here they're adding IgG, and here they're taking it away. It's like the reciprocal. Two different ways to test the same sort of like hypothesis. Okay. Now here they're testing the idea that Remember this strange symbol, this receptor? This is a receptor we know binds to IgG. So what they're testing here is plasma is complicated stuff. It's got lots of things in it. It's got many, many different things. And so they're testing, is it literally just the IgG plus that receptor that's responsible for that phenotype? Or is there something else in the plasma that contributes? And if there was something else in the plasma that contributes, you would not be able to just mix the receptor and the IgG plus the virion and get the same effect. You wouldn't be able to do that if there was something else that was necessary. Okay, so here they do that experiment. Wait, are they removing it? This looks normal. This looks normal. It's enhanced. Here they're adding. Interesting. So it looks like when they add IgG plus the receptor, it's not sufficient to produce that result. So that would suggest that there is something else that's required. And then that would explain why they go into D, E, and F, testing these other things. Well, is it this thing? Is it that thing? Is it this thing? What's the question?
For the receptor, could that be like a competition thing? Like they're adding that, so couldn't that compete with the receptor in like the cell culture?
Yes, it could. Is that how they're phrasing it? Do they actually say it like that?
No.
Or you just guess as that's a good hypothesis for why you should explain this data? That could make sense. Like in theory, like these cells already have this receptor, and if the IgG is all swapped up, bound to this one that they added.
It's a good experiment for saying that IgG binds.
Yes, I should. Okay, now I'm stuck. I should have read that more closely. Interesting. I'm looking at my notes. My notes say that they're depleting it. Let me look at the figure legend.
Well, what does BI mean? Inhibitor.
What's a binding inhibitor?
BI. That's what they're saying.
Ah, okay, that explains it. Okay, so if this is a binding inhibitor of that receptor, then that explains why they're not seeing the phenotype, because it's inhibiting the binding of IgG to the receptor. Great work, Ifan.
Oh, did you see that in the paper? Yeah. You pointed us to that.
Great work. That okay, so that explains it. Do we get that point, D?
Yeah.
Okay, so it's a
Yes.
binding inhibitor. Okay. Okay, let's go to figure 3. So figure 3, they changed the experiment. Figure 3. What do they switch to in figure 3? Mice. Okay. So this one is easy. This one's easy. Now their experiment, I've said they were all easy and they've all been pretty hard. Okay, in this experiment, what they do is they take a mouse. This is my mouse. They take a mouse and they inject it, step 1. They inject it with the convalescent plasma. Step 2, second injection with Zika virus. And then 3, what do they measure? They measure a bunch of stuff, but figure 3A, the most important figure, they measure survival. So they're killing the mice, they're injecting them with something that in theory could protect them, and then they inject them with Zika virus, and the Zika virus kills them. Now, if you guys have been listening closely to my lectures in the past, should Zika virus kill the mouse? No. Why? Like why? Give me a couple of reasons why, if it was just a normal mouse, why should Zika virus not kill the mouse? What are the reasons for that? Well, that's true. It would never, in theory, in general, would never even get the Zika virus because Aedes is not going to be feeding on mice. Although I think in the one paper that I cited in the last lecture, there's like 1% feeding rate on things that include rats. So maybe, but that's not the answer I'm looking for. Why should Zika virus not kill a mouse? Does Zika virus even kill humans? No, like Zika virus doesn't even kill humans. So like, why would it kill a mouse? Especially when like Zika virus is kind of like evolved for primates. Well, maybe there are some weird scenarios you could think of why it might kill a mouse, but like it shouldn't be killing a mouse. So what is unique about this experimental setup that allows them to measure these mice that are dying from Zika virus? Did you catch that? Is it just like a normal mouse, a healthy mouse? Yeah, it's a special genotype. It's something like STAT2 double knockout mouse. It's some special mouse that is like a sick mouse. It's already like a diseased mouse. It's got some knockouts that are clearly like important, and they've sort of picked a mouse model which they can kill easily with the Zika virus. So the first thing to just note about these experiments is these are not normal mice. These are like weird freak mutant mice that die when you inject them with the Zika virus. So the other thing you should keep in mind is like, are these experiments relevant? Like, are these experiments relevant to humans if they're only producing these data from like these freak mice that they've done double knockouts in that are already sick? Like maybe, I don't know. Maybe, I don't know. It's probably not like the best model for sure. Yeah, I mean, well, see, I like their biochemistry experiments better. The first figure 1 and figure 2, I think, are better experiments because they're using human plasma and they're using human antibodies, and they're actually testing like biochemically like human relevant stuff. In this case, now they've switched models, and because they kind of like need a model, they need something to test. So they switch to this, and this is kind of like their best option. But it's not necessarily immediately translatable to the human system. So just keep that in mind. Okay, so let's look at the figure. So survival, okay, if you inject them with just the control, plasma from normal human being, they die pretty quick, days post-infection. They die at a slower rate. So they're surviving for longer. They never die. They never die if you inject them with plasma from a normal human. They don't die after 14 days, okay? So they're all alive. If you inject them with plasma from convalescent plasma from people who had been infected with dengue virus or West Nile virus, now all of a sudden they start to die. You see that? They're dying. Every time this thing ticks down, those are some mice that are dying. Does that make sense? Okay. So why are they dying? ADE. They're dying in theory because of antibody-dependent enhancement. You've given them plasma that has antibodies, and now the Zika virus is like super able to infect the cells and it kills the mouse. Okay, so they're not dying if they don't have the antibodies. They are dying if they do have the antibodies. Okay, so this is like a theoretical mouse model proof of principle that antibody-dependent enhancement can happen in like mammalian vertebrate systems. Are mice are they? They're not mammalian, are they? No, they're rodents. Are rodents mammals? Are they? They breastfeed?
They have milk.
Interesting. Okay, I should know that, but apparently you've seen my ignorance. Okay. We got that. Figure 4. Okay, figure 4 is, I remember now. Figure 4 is, we've done these experiments with these mice. They did the experiments with the mice. Now let's like dissect the mice, and let's do microscopy to look for the virus. Where is it? Is it actually increased in replication? Is there actually more variants by microscopy? And also let's do quantitative PCR. It must be quantitative RT-PCR because flaviviruses are RNA genomes. So it must be quantitative RT-PCR to check for levels of the virus. Yep, RNA copies. So they're doing quantitative reverse transcriptase PCR on the y-axis. So if it's high in the y-axis, there's lots of virus. If it's low in the y-axis, there's not a lot of virus. Okay, and different days, x-axis is different days of post-infection. So after the infection and then longer after the infection. Okay, and again, the color coding is the same. Blue is dengue virus antibodies, red is West Nile virus antibodies, black is control. So there's more virus, there's more genomes of the virus, okay, in animals, in mice that have been injected with these antibodies from this convalescent plasma in the blood. Same results in the spinal cord, there's more virus. Same results in the testes, there's more virus. And then they just do some microscopy. Let me move this. Do some microscopy in the spinal cord and the testes. And here's the control. Here's an injection with PBS. So these should have sort of like normal amount of Zika virus. If you're injected with convalescent plasma from dengue or West Nile virus, you can see more of the Zika virus. Zika virus is green. You see more of it. Here's the testes, same experiment. Here's the negative control. You see more of it in the dengue virus antibodies, more of it with the West Nile virus antibodies. And then they check some other stuff, and I don't think these are statistically significant, but they check some other stuff like the brain, the ovary, and the eye. The main important point is the antibody-dependent enhancement effect can kill the mice, and there's more virus in those mice. That's sort of like the main points. So what are the conclusions from this paper? Conclusions. Finish up here. They actually say it. They write it out. I'll just read their words. Conclusion. Together, our data demonstrate that IgG elicited by infection with dengue virus and West Nile virus is capable of mediating antibody-dependent enhancement of Zika virus through the Fc gamma receptor engagement in vitro. So they're saying, yes, you can get antibody-dependent enhancement. Now, criticisms of this paper. My main criticisms are like, we have to be careful about trying to extract the mice models because the mice are the mutant mice. And there are those other papers that say in the introduction that it hasn't been noticed in humans that this happens, at least in the current literature that I've seen. So, and in what they're citing in this paper. So we have to be careful in sort of translating the mice models and thinking that the mice model data is going to be equivalent to what happens in humans. And it seems like it's not the case. So we're kind of probably lucky that it's not the case. But it can happen in the mice. That's kind of my main caveat. Any questions about the paper? Or questions about antibody-dependent enhancement in general?
Yeah, I don't understand the whole thing. So
Antibody-dependent enhancement, or you don't understand?
Yeah, AD. So previous infection of dengue or West Nile virus increases affinity to E protein, right?
Increases affinity to the E pro. So, okay, so let me first walk through what's going to happen. So imagine you get infected with a dengue virus 5, or maybe there isn't 5, 4, dengue virus 4. Okay, your body is going to produce antibodies that recognize the E protein from dengue virus 4. Okay, and what's going to happen in that system is these antibodies are going to be bound to the leukocytes. And in the first case, what's going to happen is the leukocytes are going to surround this virion and kill it and eat it. They're going to absorb it and eat it. Now, in a second infection, so this same person gets infected with dengue virus, let's say 2. This reaction is still good, but for some reason, these leukocytes can't kill the dengue 2.
Yeah.
You're saying that's the question? I don't understand why they can't kill it. That's a good question. What? They combine to the E protein. And what I'm saying, what we know happens in antibody-dependent enhancement is in this case, the dengue 2 gets in the leukocyte, and in this case, it replicates. And so her question is, why can it kill it here, but not kill it here? I don't know the answer to that off the top of my head. It is weird. Why can it not kill it? It probably has something to do with the mechanism of how they're getting killed. So this is a guess. I'm going beyond what I know for sure, but I'm trying to guess what I think it might be. So imagine this is a leukocyte. What's going to happen is it's going to have these antibodies, and they're going to recognize a virus. Okay. And then what's happened is once this thing is recognized, it's going to be endocytosed. So it's going to come into the cell. Okay. It's going to come inside. And this is going to get this complex, probably this complex is going to get sent to some part of the cell where it gets degraded and destroyed. So they kind of like eat these viruses. And my imagination is suggesting to me that probably getting trafficked to the place where they get degraded is probably has some kind of something to do with probably like the tightness of this interaction. Like it probably has to be very, very tight, and it probably is important in some way of recruiting other factors that lead it to get degraded. And what probably happens is in the second scenario, there's probably enough of an interaction where now you have a new virus. It's probably enough of an interaction where it can draw it into the cell through endocytosis, but then maybe once it gets into the cell, it's able to kind of like break off. Does that make sense? And it probably doesn't end up getting trafficked to where it gets killed. That's my guess for how mechanistically it happens. But you guys should fact check me and maybe come back and you guys should present the next lecture and describe the mechanism. That's what I would guess. Any other questions? Yeah.
So in this paper, they use mice, specific mice, and why they cannot use normal mice? And
because normal mice will not die from Zika virus, so there's nothing to measure.
But after they get pregnant, they have the baby, like the human.
Do mice get microcephaly?
They can
I don't know, but even that's going to be even more of a pain to test because you're going to have to like rear pregnant mice and like infect them with the Zika virus like when they're pregnant. Maybe that wouldn't be too hard. I don't know. I don't know if there's pathology in mice who are pregnant who have Zika virus. It's probably, I don't know. I don't know what the models from microcephaly are. That's a good question though. You should present on that.
No more questions?
Okay. All right. Have a good Friday. Everybody have a good weekend. See you on Monday.